Today, I'm delighted to welcome Cesária Victoria, who's our speaker for today's webinar. Cesária is a Mozambican early career young scientist. In 2024, she defended her master's project focused on social vocalizations of insectivorous bats in Gorongosa National Park, which represents the first study revealing 11 different types of social vocalizations for Hipposideros kaffler and adding 4 new different types of social vocalizations for Nycteris debaica. Her current professional activities include scientific research on bat social communication, monitoring bat populations, and tracking post-guano extraction recovery processes at Gorongosa National Park and its buffer zone. Cesária also provides scientific support to Limpopo Watercourse Commission, which is a transboundary river basin organization conducting a project aiming to achieve integrated cross-sectorial ecosystem-based management, uplifting the living standards of the basin's population and conserving basin's water resources and ecosystem services in the Limpopo River Basin. In addition, Cesária is a member of Bats Without Borders, Bat Conservation Africa, and Global South Bat. And after participating in the Bat Group's training organized by Bats Without Borders, Cesária, along with other Mozambican conservationists, formed the Maputo Bat Alliance in Zinave Bat Alliance, aiming to promote environmental education activities focused in raising awareness about bat ecology and conservation. So today she's going to be talking to us about social vocalizations of insectivorous bats in Gorongosa National Park, and we are very much looking forward to hearing more about that. So I'm going to stop sharing my screen and hand over to you, Cesária, for your presentation.
So as Elisa was saying, my name is Cesária. I'm a Mozambican bat biologist, conservation biologist, and today I will be talking to you about my research, master's project research about social vocalizations of insectivorous bats in Gorongosa National Park. Oh, sorry. As we know, communication is a very important behavior among many different animals, and it's a characteristic that can guarantee the survival and reproduction of different species. And for bats, many different behaviors related to communication have been studied through time. And one of them is exactly the ability to use sounds to be able to convey different information about what is happening around them. Different sounds or vocalizations to be able to identify individuals conspecifics, as well to identify the pups when females come back from foraging, and to determine their territory, and also to court females during the mating season. We can see in the image, for example, for one of the species that I was studying, in the left, we can see an individual male possibly courting a female, and on the right, we can see a female interacting with her young pup. In this video, we can see also an interaction we will see an interaction between individuals of another species that I was studying, Nycteris debaica. What we are hearing now are the different social calls that they produce while in roosts, while in the beginning we were hearing their echolocation calls. So in general for birds, we are more familiar with echolocation calls, but birds produce two different types of acoustic signals. They are echolocation and social calls. Echolocation calls are predominantly used to navigate and to detect prey, while social calls, they are primarily used to convey social information. But there are studies that have been describing also that have been associating the echolocation calls also to the possibility of convey social information like identifying individuals, roosting sites, roosting mates, and different others. But in general, echolocation calls are mainly used for navigation and detection of prey. It's possible to differentiate echolocation and social calls when looking to their sound characteristics in sonograms. Comparing the spectrotemporal characteristics that we can observe. So for example, in these sonograms, we can see in the left, the echolocation cloud, and we can see a very simple single type of call, while in the right, the social call, we can see a much complex vocalization. It presents different syllables, and we can also see that the social call presents a much lower frequency when we compare to the echolocation call of the same species. The social calls can also be distinguished in calls, which are less complex calls, usually short, and even though they can present some repetitions, that is not usual. And calls are used for many different functions, to determine territory, to courtship, to identify conspecifics, to identify roosting sites, and many other functions. While when we talk about social songs, we are looking to a more complex type of vocalization, which is usually repeated in stereotyped, and it presents a much complex temporal and spectral structural characteristics when we compare to the social call. And the social songs are usually only produced for territoriality or for courtshipping. Although birds present very complex characteristics, I'm talking about their physiognomic characteristics. They are very diverse. The social calls are the social calls, and specifically the social songs, are only known for approximately 20 species of birds. Even though the social calls are much known than the social songs, we currently know very little about the social vocalizations of birds. The studies that have been published have been showing that even though the birds are very diverse, and they can present very diverse social vocal repertoires, some vocalizations can present some similarities. As we can see in this study, for example, the authors could show that these two species, they present some social calls that present similar characteristics. But when we compare the temporal characteristics of each vocalizations, we can see that they are species and individual specific, which means that we can distinguish the social vocalizations according to each species, even if they present some similar characteristics when we look to them. And when we talk about a specific type of call that we call distress call, which is a call that the birds produce when they are caught by a predator, we can see that the distress calls in particular, they present a much similar structure when we compare them to different species, which suggests that the distress calls are possibly used to elicit conspecifics and other species when they are emitted, probably to mob the predator. So during my study, I was trying to see if the social vocalizations are really predominantly used for intraspecific communication. If it is the case, so they will present more spectrotemporal variation between species than within species. And if these distress calls are used to elicit responses from both con and heterospecific individuals, they will present similar spectrotemporal characteristics across species. My study was conducted in Gorongosa National Park, both in the core and buffer zone areas of the park. I had two focal species, Epositorus scapher and Nycteris thebaika. For Epositorus scapher, we didn't know any social vocalization. We only knew the echolocation call that is presented here. And for Nycteris thebaika, a study done in the 90s could present two types of social vocalizations for these species. So one of them described it as a contact call. I also had some additional species, Scotophilus viridis, Pseudorhynchus rindalai, and Afronycteris nana. All of them belong to the same family. And that was a very good opportunity to also explore some other species within the Gorongosa National Park bird richness. I collected data both in the roosts of my focal species. These are some of the roosts that we can find Nycteris thebaika in Gorongosa. As well, that abandoned building is one of the roosts of Epositorus scapher in the park. And I also collected data in the lab. So I would set recordings in the roost, but also collect some individuals and take them to the lab to record them. And after 24, 48, or 72 hours, release them back to the roost. I collected or I recorded individuals using the Wildlife Acoustics SM4 bat recorder, as well Z infrared camera to make the video recordings. And so both the video and audio recordings were synchronized to be able to describe the behavior according to the sound. And to conduct my analysis, I use Kaleidoscope pro-analysis software to run a cluster analysis, allowing me to cluster similar recordings. Then I use a Draven to be able to measure the spectro-temporal characteristics of my recordings, to be able to describe the spectro-temporal characteristics. And I was basically measuring the frequency and the duration of the calls. I also used QuickTime Player and Audacity to analyze my videos, and the Audacity specifically, I use it to be able to extract the video from, to extract the audio recordings from the video, allowing me to synchronize the videos and the audios more easily. And for my statistical analysis, I run a PCA in the Euclidean distance. So the PCA allowed me to cluster the interrelated vocalizations, the one that showed similar characteristics were clustered together. And the Euclidean distance, I use it to measure the similarity between the calls. So the higher the Euclidean distance, the lower the similarity. So for a procedure cover, I was able to identify 11 different social vocalizations. I'm not showing all of them during this presentation, but for example, this one was one that I suggest is a contact call. I could find them both in the roost and in laboratory captivity recordings. In yellow, you can see the call duration. In red, the fundamental harmonic, and in green, the second harmonic. So this one is possibly a contact call because I could find them both in roost and captivity recordings, and they might be used to identify or contact conspecifics. And I suggested that is a contact call exactly because I could find also in roost recordings when I had just one or two individuals that probably were trying to identify some roost mates. And these bats are very, they are highly gregarious. They are usually found in very big groups. I also highlight here a possible song, courtship song. This recording is in structure complex, it's low in frequency, and songs are usually much low in frequency when compared to calls. In this case, I suggest that it's a courtship song because I could have some video recordings of individuals, especially during the mating season, where I could see different interactions during the mating season. And I could not find any other vocalizations that I could suggest that is a territorial song, for example. And when the individuals, for example, were interacting aggressively, possibly to demark their territory, they were not producing any song. And this is possibly a song exactly considering the complex structure that it presents. First of all, it's very repeated. As we can see, the similar structure of the syllables repeated in the call. And as well, it presents two different syllables in two different frequencies, which highlights how complex the vocalization is. For Nictoris Baiga, I could identify 6 different types of vocalizations. This one is a contact call, which was also identified and described as a contact call by Aldrich in his research in Zimbabwe in the 90s. Although we find the same type of vocalization, we describe our vocalizations in different frequencies and duration. These differences that we are observing can be an indication of geographical variation of this call among different populations of the same species. And when this was one of the vocalizations that I could synchronize with video, and I could see that when one individual was approaching the other in the lab, the individual approaching the other would start emitting this vocalization. And in the paper, the author said that when one individual was in the tent and the other outside of the roost, the individual in the tent was producing one vocalization and the other individual outside of the tent was also producing this vocalization that he could identify. Both of us, I mean, myself and the author of the paper of the in the 90s, we could identify these type of vocalizations that we could not describe according to the behavior, but again, we see vocalization described in or presenting different, slight difference on its temporal characteristics, which again can be an indication of geographical variation. For Nictoris Baiga, I also identified a possible territorial song. I suggest is a territorial song because I could not observe any courtship behavior during the period that I was doing my, that I was collecting data. And I suggest that is a territorial song, also observing the lower frequency that it presents, and the fact that presents repeated syllables with a very complex structure. We can see here where I highlight in red that these syllables that apparently are very similar in structure, they end in very different ways, which highlights how complex these calls and the information that they convey, how complex these calls are and how complex can be also the information that they are trying to convey. When we look to the distress calls, I could collect distress calls for the 5 species that I have. I mean the focal species and the additional species that I could record. And we can see that the distress calls for all of them present some similarities, especially because all of them present this bus syllable, which is highlighted here for Afrenitis, Pseudomyces cervidali, as well for Episidurus cover. But we can see that the bus syllable is present in all, or in each one of these recordings that we can see. And for some of them, I could also record the tonal part. So the fact that these vocalizations are present some similarities between them they are an indication that the distress calls in different species present more similarities when we compare other calls between different species. When conducting statistical analysis for Apposideros cover, all the vocalizations they cluster among them, and the Euclidean distance varied between 2 to 95, which means that there are calls that are more similar between them, and there are calls that are more different. But in general, the calls of Apposideros cover, they present temporal similarities between them, which means that they are used to convey information among conspecifics. And the same we can see for Nycteris the biker, the Euclidean the distance between the calls, it's even lower, which means that the vocalizations present similarities among them, But when we compare this, the same type of call, I mean, a contact call identified for Nycteris the biker and the contact call identified for Apposideros cover, we can see that they do not cluster. They are completely separated or in different quadrants, which means that these contact calls, even though they are described for the same context, which is contact, they are completely different between them, which means that when they produce contact calls, they are only used to be able to locate or identify conspecifics of Apposideros cover or Nycteris the biker. And the Euclidean distance we can see was the higher, was higher when we compare to the Euclidean distance that we saw previously. While when we compare distress calls, which are also the distress calls of the 5 species, even though we are comparing calls of different species, we can see that the Euclidean distance it's lower when we compare to the contact call of Nycteris the biker and Apposideros, which means that the distress call, even though it's a call that is produced it's produced by different species. They present similar spectrotemporal characteristics, which means that they are probably used to convey information not only for conspecifics but also for different species. So, in general, or as a consideration, social vocalizations are largely used to convey information for conspecifics, so they are species specific, but the distress calls in particular, they can be similar across species. So they can be used also to share information between different species. To our knowledge, this is the first time revealing the vocal repertoire of Apposideros cover, and we still have a lot to describe and discover. And this study can represent the first revealing social songs for 2 families that we don't know social calls, which are Apposiderida and Dicterida. And through this study, there is a possibility to use social vocalizations to monitor bird populations. with special attention to Nycteris thebaia, which is a species that it's very difficult to identify in the field through the echolocation calls, because the calls are very quiet. So it's important for monitoring purposes that we can identify Nycteris thebaia through their social vocalizations. Each of these species present complex social behaviors, so it's not only about their social vocalizations, their social behaviors in general, looking at olfactory, tactile, visual communication. They they present a lot of information that it's still needed to be properly documented. And I also recommend for future studies that individuals should be marked, so it will be much easier to describe specific behaviors of males and females. So this is a brief about my master's project. I am very grateful to have the opportunity to be part of the master's students of Gorongosa National Park, of the Bioeducation Consortium of Gorongosa National Park, which is not only Gorongosa, but it's also Universidade de Lúrio, Universidade de Zambezi, Instituto Superior Politécnico de Manica, and Universidade de Lisboa. As well, I'm grateful to the HHMI because they were the main institution that provide the fellowship for us, and to the the Nature, Environment, Wildlife Film organization, which is a South African organization that is working with scientists across Africa to document and yes, to document their research, and also bringing many different interesting research about wildlife and communities, wildlife and communities biodiversity. And during my my master's degree, I had the opportunity to have two documentaries produced by new team, The Voice of Birds and the Guano Gold. They are available in YouTube, in the Wild Hope channel, as well in the New channel. So if you're interested, you can check. And The Voice of Birds in particular also talks about my research. As well, I'm very grateful for the different articles that are published about my work at Nautilus and Merlin Turtle Bird Conservation, and recently in Nature. As well, I am grateful to the super scientists that it's a South African organization as well that is promoting African researchers using their research, highlighting them as superheroes. And I'm also very grateful to my supervisors, to the entire GRP team, which is Gorongosa Restoration Project team, specifically to the scientific department of the park, and my family, friends, and everyone that have contributed directly, indirectly to the success of my project. Well, this was about my research. I'm now going to talk a bit about Mozambican Bat Alliance. So, Mozambique Bat Alliance is a vet group from Mozambique. We are basically three. Currently, we are only three, and we expect the groups to grow. And we have started making very good contact with different researchers in Mozambique that are interested to join. So the Mozambique Bat Alliance is a bat group which is trying to promote the bat conservation through environmental education, and is also trying to connect bat researchers and enthusiasts in the country. We are part of Bats Without Borders, and we were part of the first Bat Groups training. So through the training, we started the Mozambique Bat Alliance. So we work basically where we are based. So I am based in Maputo, and Aglet is based in Maputo. So we conduct some bed talks or bed activities in Maputo. Osvaldo is based in Senave National Park, so he conducts some activities in Senave or in Yambani province. We could through the activities that we could conduct, we could basically reach almost 500 people in the country in different parts, especially in Yambani province, which is Senave National Park and Mashishi City, and also in Sofala in Gorongosa National Park, through different activities with interns, with students, master students in the park, in primary and secondary schools. And we are also planning some more activities in Maputo, in a primary and secondary school, as well. Vet survey, the first, not well, I cannot say the first, but will be a new vet survey at Senave National Park. You can also, if you are interested, join us. As Elisa was saying in the beginning, I am part of the Bats Without Borders. I'm also part of Bat Conservation Africa and Global South Bats. In the beginning of the year, I had the opportunity to join the Global South Bats in Malaysia to the second networking. That was an amazing experience, and one of the main results of that networking in Malaysia was the development of a cross-continental project. And in my group, we are talking about, we are developing a project about a bad suppression in coffee plantations. So this is a bit about me and my work. I hope you have enjoyed, and I'm happy to receive questions. Thank you. Muito obrigada.
Thank you, Cesária. That was fascinating. And I just have to say, I love the cartoon of you as a Batwoman. So, great. So it was great to hear, obviously, about your research on bat vocalizations, but also great to hear about your experience with the Mozambican Bat Alliance and the outreach and education work that you've been doing. So thank you very much for sharing that with us. We have plenty of time for questions, and we already have a couple of questions in the chat, so I'm going to start with those. So we also have a couple of comments saying fantastic study and great presentation. So I'm going to start with a question from Elise, which asks whether there were many types of calls that could not be attributed to a specific behavior.
Yes, I still have a lot of calls that I cannot yet describe according to a specific behavior. For example, I said that I could identify 11 different type of vocalizations for a procedure cover, and I suggest at least, or I could at least suggest that some of them were courtship song or courtship song or contact calls. But for the other 9 calls, I still have to collect more data and try to synchronize. The thing is that the challenge that I face is to have very good interactions in captivity, because in the field or in the roost there are too many individuals. The camera is recording many different individuals, and the batch recorder is also collecting sounds from everywhere. So in captivity, it should be easier, and it is actually easier to focus in very small number of individuals. But in captivity, they do not produce always the same type of vocalization that they produce in the roost, on their natural roost. So it should be necessary to keep them in captivity for long or for much longer periods, and keep them habituated to that environment, so they maybe by then they will start behaving naturally. But that takes time, and it's also about dealing with stress of each individual, so it's a process.
Yeah, because presumably so it's easier in the lab, but you also have fewer individuals and fewer interactions. So that limits your ability, presumably, to identify those goals. And there was a follow-up question also from Elise, asking if you detect more distress calls when you were recording them in the lab. But I guess that goes back to there's fewer individuals, fewer interactions, but they're in not a natural environment. So yeah, I don't know what that means in terms of distress calls.
The distress calls, I usually recorded them when I was handling them. So, for example, when I set nets to try to catch them, when I am pulling them out of the net, I record them that right in that exact moment. And in the roost as well, because sometimes I had to go to the roost and try to collect some individuals. When I was handling the individual, I would record them. So in the roost or in captivity, I didn't find distress calls after releasing the birds, because also considering that usually distress calls easily recorded when the bird is caught by the predator. That's why I had to record the distress calls when I was handling them, because when they are free, they probably will not produce the distress call, as far as I know.
Great. There's more questions in the chat. Question related to background or interfering noise and how you handle that.
I usually when conducting my cluster analysis on Kaleidoscope, I usually selected the calls that did not present that much background noise to not interfere on my analysis. So that was the first thing. Just try to only conduct analysis on the calls that did not present that much background noise. And if that is the case, when presenting the sonogram, I will try to exclude the background noise. But sometimes I have, like for some calls, I don't have that much recordings, and I had to deal with the background noise, trying to exactly see what is the extent of the background noise, and see as much as possible how I could exclude it.
Great. There's also a question from Cecilia asking, do you know how the social vocalizations of your two focal species compare to other species in those genera? And if so, so are they different from other species within the same genera?
Good question. And also, that is something. No,
just asking if those social calls can be helpful in distinguishing between species within the same genera.
That is a good question, and that is something that I'm trying to do. So I don't have a full answer right now, but I'm currently also conducting some social calls recordings in some of the colonies for some of the species. One of them is from the same family. I still have to find more colonies with the same gender. So yeah, right now I don't have the answer, but yes, I am trying to find it. I remember that I had for some occasions the same gender for Nycteris. I had Nycteris the pica and Nycteris grandis, and there are some similarities in some vocalizations, but I don't have that much recordings to be able to see it perfectly. So I'm trying to find more individuals in the colonies to be able to do it. That's why I was saying in the beginning that we have so many species, and in the African context especially, we know even less than for European species, for example. So we still have a lot to do in terms of documenting the social vocalization of different species, so we can actually use this knowledge to be able to monitor different populations. But yes, Cecilia, that is something that I'm also trying to see.
Lots more to learn, much more research to do. There's also a question about Bat Alliance, the Mozambican Bat Alliance, asking about the criteria that you use to select the schools and about the age of the children that you were reaching out to.
Well, right now, first we try to like knock the door where they open the door for us, so we can work in that schools. And it depends on the availability of the school, of the teachers, and in terms of age, we try to work at least from the 30 or 40 degree. So yeah, like starting from the 3rd or 4th degree, because they can understand a bit what we are saying. They can even in the 2nd or 1st degree, they could paint or things like that. But from 3rd degree, they already have a natural science, so they have some knowledge about what is nature. So it's a bit easier to introduce some new contents also about beds, for example. So we try to focus from the 3rd or 4th degree.
And then you go up to, you mentioned also secondary schools.
Yes, we go up secondary schools, universities, and yeah.
And you
during the training, the Pets Without the Bed group training with Pets Without Borders, we had very different contents, and one of them was School Without Walls, and we were inspired by how they were able to create different contents, also using cartoons to be able to talk about pets. So we also use these contents that are produced exactly for children to be able to go to the schools and introduce pets for children.
And do you find that there's any difference in how receptive the kids are to what you're saying about bats? Is it younger kids are a bit, you know, more excited or more receptive to what you're saying, or is it the other way around? Maybe older kids that have a bit more knowledge about nature already, perhaps they're a bit more open to finding out more about bats in this case.
In general, I feel the kids or children very open to learn. They get fascinated. They want to. They start making many questions about pets because it's something that usually on the daily basics they don't have. How can I say, they don't have access, they don't see pets easily. So if I go to them and I say that, oh, did you know that pets also communicate between mother and babies? They will say, huh? How can you do this? How do they do that? And many different things. So if I say that pets are pollinators and bird eats insects, so children in general, they get fascinated easily when compared to adults, because adults, for example, if I say them that birds are important because they are pollinators, so they eat insects in your crops, they will start saying, no, that's not true. I had the birds that were destroying my crops of bananas. And yeah, we have to be, how can I say, I will not say more careful, but I have to work a bit more with adults to explain that it's actually the other way around.
Yeah. Yeah, no, that's very understandable. And that actually links to the last question that I'm going to ask, comes from Karen in the chat. And she asks, what are the biggest challenges in creating groups focused on bat conservation and implementing activities? So that's obviously a very big question, but maybe if you can base it on your experience, that would be really useful.
Well, right now, I can say that the main challenge is keep consistency and dedicate time to that. Because there are many activities, like, for example, right now we are 3 in the group and we expect the group to grow. And Karin, for example, is one of that person that we expect him to come and join us. And we need to dedicate time and try to make sure that, yes, we keep consistent. So if we have this activity planned, we have to do the best to really make it happen. Yes, I think like the first challenge is that consistency and time to do it. And then we will the challenge, it's possibly, or at least what I am facing, it's to have as for example, when targeting a school, to have the door open, because the school will take time to respond and they will say that we don't have time or something like that. So it's something that sometimes can also takes time when talking about targeting a school.
Yeah, but it sounds like the work you're doing is really amazing, and I'm sure will be having an impact on the children and kids that you're speaking to. Right, we've run out of time, but just wanted to thank you again for sharing your research and your outreach experience with us, and also thanking everyone else for joining us and for asking lots of interesting questions. So I hope you'll continue to join us in upcoming webinars, and I hope you enjoy the rest of your afternoons. Thanks, everyone, and thanks again, Cesária, for sharing your work with us.
Thank you. I hope you have enjoyed.
Yes, we all did, I'm sure.